Hi kids, it's Mrs. Fravel. How are you? Good. Me too. Hey, we are about to finally begin talking about the specific groups of animals in the animal kingdom. We're going to start with the animal phyla groups. Today we're going to talk about phylum Porifera. We're going to talk about sponges. This video is going to be a little long because if we were in class, Porifera would take us 2 days. But with the whole pandemic shutdown, shortened year, truncated stuff, I'm going to squish it all into this one presentation. But the good news is that your assignments are pretty short as well. Your only assignments for this week are to take notes from this video in your notebook, and that includes drawing pictures and diagrams of sponge body morphology, which will be later on in this video. And then you're going to upload those. You gotta show me proof, take pictures of your notes and upload pictures, and then your Prezi for Porifera, which I will explain at the end of this presentation. So without further ado, let's talk about some sponges, shall we? All right. Sorry about that. I had to go grab my sponges. I forgot them. So you should have already done these starter questions on Canvas because, of course, you are doing this properly, where you go to the home page and then you go to the modules, right? And so the starter question should have been the first thing to appear in 2 modules on phylum Porifera for this week. So let's go through the answers really quickly. As a review, what are the 3 forms of animal body symmetry? That's asymmetrical, radial, and bilateral. Remember when you did the candy lab and you had to cut up all those gummy candies? That was that, okay? 3 levels of organization in animal bodies. Remember, everything has cells. Then the next level of organization, cells work together to be tissues that perform a specific metabolic function. Tissues get together and create organs that support a life function, and then organs that do similar functions work together in an organ system. Okay, so depending on the group of animals, you'll see different levels of organization. Today we're going to be talking about an animal that's just at the cellular level. I'm going to try to not let my mouth get ahead of my brain today. So that leads to the next question. Let's see if you figured this one out. Physiologically and structurally speaking, what's the simplest animals? That's right, it's sponges. And that's what we're going to talk about today. We're going to talk about phylum Porifera. Woohoo! These are the simplest, oldest animals. These were the first animals to appear in the fossil record on Earth and the first multicellular eukaryotic protists working together to create a body. Phylum Porifera has been around on the planet, as best we can tell from the fossil record, over 500 million years, over 500 million years. And they started the explosion of biodiversity that is now what we see in the animal kingdom through time and what we see around us alive now. They are simple, but they are perfectly suited to their environment, and they have not changed much at all as a group over the last 500 million years. Now, of course, sponges all are the default animal. They were where the rest of us come from. Genetic mutations arise, right? That's how evolutionary progress happens. Mutations happen. Those mutations sometimes result in a new structure or new metabolic adaptation that is better suited to an environment, and that is what leads to speciation. So sponges are still here because they still work great. The rest of the animal kingdom evolved from those original sponges over the last 500 million years just via the miracle that is natural selection by mutation and evolution. Okay, so we have there's a lot of good diversity just within this phylum. We get sponges that range from tiny sponges to sponges that are bigger than humans. There's been sponges discovered in the deep sea that are larger than a minivan. We get these beautiful vase sponges. These are glass sponges. I'll talk about those in a minute. We get these funny tube sponges and encrusting sponges, and we get tiny ones. We get big ones. You guys, they're great. Look, I have sponges. These are two different kinds of sponges. I have several. I also have a couple of pet sponges living in my reef tank, and they're just, you know, super chill. Okay, so what is a sponge? A sponge is an animal that has these characteristics, basic characteristics, and this is what you'll put on your Prezi. All are sessile as adults. Their larvae are motile. They have indirect development, so they have a larval phase. We call that the planula, and then they metamorphose into an adult sponge. Okay? They have asymmetrical body plan. They're the only animals that do. Remember, that just means that while they might have a morphological plan for their bodies, which I'll talk about in a minute, there's no guarantee that if you cut them in half or if you cut them into different parts, you'll ever come up with two mirror images, right? If you cut you in half, you got a left side and a right side that are the same. Sponges don't do that. They're asymmetrical. They can go, one side of them might be big and bulbous and the other kind of squishy flat, and another member of the same exact species might have the opposite. They might be squishy all over, tiny. Okay, so it just depends on where they are living and the available nutrients, water parameters, that kind of thing. Okay, so asymmetrical, no body plan. And then they are, again, super simple. They are at the cellular level of organization. They're the only animals that have this. They have no tissues, they have no organs, they have no organ systems, they have no mouths, they have no brains, they have no eyes, they have no nothing. They are just 4 different kinds of cells that live together. Okay? Now, a little bit about the terminology and some new vocabulary. The phylum Porifera, that name Porifera just means pore bearing, and that is because the external surface of their body, actually their whole body, is covered with holes. They're covered with pores, and you can see them, okay? There's 2 different kinds of pores. The smaller ones are called ostia. The larger ones are called oscula. One is an osculum. Okay, so those function as the animal's inflow and outflow for their body, and I'll show you that in just a sec. So let's talk about what they are made of. Their cells themselves, they have 4 kinds. That's it. They have 4 different kinds of cells. Their machinery, like the most active kind of cells that they have, there's 2. One of them is their choanocytes or collar cells. And these line the interior of all of those little canals. So when you have these holes, the little holes in them, inside of those, the cells are choanocytes that line the inside. What they have is a little tail. I'll show you a picture in a minute. So they have a little flagellum on each of those choanocyte cells, and they all start to whip, and they create a current that pulls water through the sponge's body, through those canals, and that current of water through the body constantly, that is what will help the sponge do gas exchange. Okay, so they get fresh oxygen, they get rid of their carbon dioxide through that flushing of water. It also brings in their food particles because these guys are filter feeders. Okay? There's also a collar. So there's a flagellum tail, and then there's this nice little collar. If you think of a cone of shame for your dog or cat after they've been to the vet, where they have that big Elizabethan plastic collar. So these cells have that as well. They are sticky and they trap the food particles that come in through the water. They also will trap sperm from another sponge. Excuse me. So that's choanocytes. The other types of cells that they have are pinacocytes, and these are flattened cells covering the external surface of the body. It's like their epidermis or their skin. They have porocytes, which these are cells that surround a pore or have a pore through them, and that's what helps form those ostia where the water comes in, okay? And the amoebocytes, these are kind of weird. These are like little amoebae that run around inside the cell's body, and they do all kinds of stuff. They're like a primitive immune system where they will they'll surround and phagocyte, which is surround and ingest bacteria or viruses that have invaded the sponge's body. They also help out with some digestion, moving around of stuff. They clean up garbage in the cells, in between the cells. So and they help with reproduction as well. These guys they're weird, but they're there. Okay, so just four different kinds of cells. Make sure you know what each of those cell types is and does. Okay, so here's some illustrations of those cells. Okay, so this is one type of cell, or I'm sorry, sponge body. This is an asconoid. Yeah, asconoid cell, or oh my gosh, I'm fine. Because I already recorded this video once and it tanked. So, sorry. This is an asconoid body type for a sponge, and the cells are in this cutaway. So in the sponge's body wall, I'm fine. I'm fine. In the sponge's body wall, we have those four kinds of cells throughout their whole body. So we have the choanocytes that line the interior of the canal and this inner chamber. And here's what those choanocytes look like. Okay, they've got a nucleus in them, just like every other cell. They have these little roots that anchor them together and to the other cells in the body wall. Here's that flagellum that'll whip back and forth. That creates the current, and you get all of them lined up like this. It really starts to pull water through. And then their collar right there that kind of surrounds the flagellum, it's sticky, so they trap food particles. Okay? The other cell types they have, here's that pinacocyte. You can see the nuclear body, and then it's kind of this squishy shape. That's why animal cells are so great. Animal cells can take on all kinds of shapes, right? And the porocyte cell creating the ostia, the actual inflow, the hole and pores in the sponges. And then the funky wandering amoebocytes doing all kinds of stuff. Okay, so here you can see there's one type of sponge body, asconoid or synconoid. Those are the vase ones. They have one big internal opening. They look like a vase. Or a tube. And then the other one, this is a leuconoid, where they have a lot of little chambers inside their body, and all of those little chambers are lined with choanocytes, and then the pinacocytes are just on the outside. Okay, all right. So how does water flow through a sponge? As those collar cells are whipping their little tails like crazy, those flagella are beating and beating. The current of water flows in through the porocyte, so it goes in through the ostia into the canal, and then there are internal chambers in the sponge, whether it's one big one or lots of little chambers. We call those atria or spongiceal. Okay, so that's that middle open bit. That's where the gas exchange is going to take place with oxygen and carbon dioxide. It's where they're going to be getting rid of their metabolic wastes as well, just cell-to-cell diffusion. And then the water flows out, up, and out through the osculum. And again, there's either one osculum, like in this picture, or in my little friend I have here, this guy's a leuconoid sponge, and I'll talk about what that means in a minute. He's got a lot of smaller oscula, and you can see those as the larger hole. And if I kind of open it up, you can see inside he's got those many atrial spongiceal. Okay, again, they're just all over the place in this guy, everywhere. Guys, this is technically a bath sponge. Did you know that that's what bath sponges are made of? There's going to be a video linked under this one for you to watch as well about that. Okay, again, there's those beautiful collar cells and amoebocytes. And when they catch food, phagocytosis of food particles. So when they catch food, what it does is that collar will move those food particles to the cell's main body of just that collar cell. And then phagocytosis is when the cell membrane surrounds something outside of it and pulls it in. And so it goes into the cell and then it's moved on to an amoebocyte where it will be digested. The nutrients will be broken down and then it starts to digest, it starts to diffuse from their cell to cell through the whole sponge body. Excuse me, got the hiccups. Okay, so how do they do that? Feeding and digestion in a sponge is super easy. They don't have a digestive system. There's no mouth. This is not a mouth. It's an osculum. And here we go with the memes, you guys. I love the memes. They just digest once they have absorbed, caught some organic matter like plankton or whatever it is. The cell digests it and then they just pass the nutrients across their cell membranes from cell to cell all over the body. Just whatever cell catches the food, they share it with all of the cells around them. Okay, and these guys are filter feeders. They just, as the water is moving through their body, through their ostia, spongiceal, and out the osculum, whatever's trapped in that, whatever comes in with that water, if it's food, they will eat it. Plankton, no problem. Tiny larval crustaceans, you betcha. Just random organic large molecules, yes, also that. They are a great vacuum or a great filter for the ocean, okay? Okay. Whew, what else? These guys don't move unless they don't move once they are adults, right? They are mobile only as larvae. So their larvae look like this. They're called, I think they're called planula. I know they are in cnidarians, and I'm suddenly blanking as to if that's right. But their larvae are this little ball of cells that has the flagella on the outside, so it can kind of maneuver a little bit. It's carried for a few days in the plankton, in the water currents, and then it settles out and then begins its metamorphosis into an adult. So again, these guys have indirect development, and then once they're settled, that's it. They don't go anywhere. Reproduction, they can do both asexual and sexual reproduction. Their asexual reproduction comes in the form of budding, and they essentially they build a little clone of themselves. You can see this sometimes on plants as well. If you get new tiny baby leaves on the edge of a leaf of plants, that's a clone. That also happens in sponges. We call those gemmules, and then the gemmules will kind of break off, be released into the water. They settle. They don't go through metamorphosis because they're already just a clone of the adult. So they're a full adult already. They fall off and just continue to grow wherever they settle. Okay? Sponges, great at regeneration as well. You can make more sponges from one sponge. If you have a live sponge, you can cut that sucker up any way that you want. Throw it into the ocean or into a tank with the right, water parameters and nutrients, and you'll get new sponges growing. You can put them in a blender. You could puree a live sponge, which, I don't know, kind of sounds awful, right? But remember, they don't have nerves, they don't have brains, they don't have an existential crisis about, oh my God, I'm in a blender, right? So you can put a live sponge in a blender and then pour it into the water, and you'll get a bazillion new sponges growing just from that. Okay? But they also, of course, just like any animal, they also produce gametes and they reproduce sexually. They are all hermaphroditic monoecious. So every sponge has both male and female gamete production capabilities. The sex cells are made by the amoebocytes. So those amoebocytes do anything. They make the sperm and eggs. The sperm are released from the sponge into the water, out through the osculum. Okay, these big clouds of sperm. And they float in the current, and if they happen to bump into another sponge of the same species, the sperm will enter that sponge and be used to fertilize the egg cells in that sponge. So they generally don't self-fertilize. They just release sperm. Then sperm from another sponge comes into them, fertilizes their eggs, and they go through that blastocyst, blastula phase, zygote, blastula, and gastrula, and then they'll be released as the larvae into the ocean from there. Okay. They don't really have a skeleton, but they're not too squishy. So the body wall of a sponge is kind of made of this protein called spongin. They named a protein after them. And then also they have these little supports called spicules, and these are microscopic crystallized silica. And also there's one group that has calcium in their spicules, and they're kind of pretty. They're very pretty. These give them some support, but also protection. Sponges aren't delicious to eat, and if you hold a sponge, they're kind of crunchy, and that's the spicules crunching in there. Okay. All right, I am going to link this video underneath this video for you to watch. Make sure that you watch it and take notes. This is Jonathan Bird. He's my secret nerd marine biology crush, you guys. He's the most enthusiastic marine biology guy I've ever seen. So enjoy, okay? Okay, I will move on. Okay, usually this would be day 2 of Porifera, but again, cramming it in so we can get to everything. Some reminders from the last unit. Morphology is the physical shape of a structure. Physiology is the metabolic function, how that structure functions. And then taxonomy, that's how organisms are grouped according to their physical characteristics. And remember, kingdom, phylum, class, order, family, genus, species. We are talking right now, I have been talking about phylum level. Okay, so again, we're in kingdom Animalia, phylum Porifera. I'm going to talk now about 2 things. I'm going to talk about the 3 different kinds of morphology that sponges have. I already just talked about their physiology. And then I'm going to talk about their taxonomy, the 3 classes of sponges, okay? So their general morphology, and here's where you, on your notes, get to start drawing. So you're going to pause me, and you're going to draw what I tell you to draw on your notes, okay? So I would like you to draw one or both of these diagrams. These are like a cutaway of a sponge body. And let's talk about their anatomy a little bit. So they have their incurrent pore, their ostia. They have a central cavity, that's the spongiceal, and then they have their osculum, okay? And they have generally more than one, always more than one ostia, sometimes more than one spongiceal or atrium. Some of them have one osculum, some of them have many. So go ahead and pause me and draw that, okay? Sorry, are you back? Okay, great. So then on that drawing, what you should also draw is the path of water through a sponge. So that was back also a few slides ago, but make sure that you get it drawn on your notes at some point. So make flow of water in and out, okay? All right, here's where you're gonna do more drawing. I'm gonna go right there. There's 3 kinds of sponge morphology. They take 3 different body shapes depending on the class, okay? The first morphology is called asconoid. Sorry guys, I'm gonna just get a quick sip of water. Very parched. Okay. Asconoid sponges. So you should pause me and draw this. Are you back? Okay. So asconoid sponges are the simple vase-like structures, and these guys are a little bit smaller. I'm gonna grab this and show you guys. I wish you guys were in class so I could pass sponges around. Okay, so these are some different sponges. This one right here is an asconoid morphologically, I believe. If there's a professional sponge scientist, please correct me if I'm wrong. This is an asconoid, I believe, and the rest of them are not, okay? So these are simple vase-like structures. They have one oscula at the top, one osculum at the top, covered with ostia. They've got that one spongiceal in the middle. So again, you should draw that and draw the flow of water. Make sure you label the ostia or incurrent pores, atrium, osculum, okay? Okay. Second one is called synconoid. And synconoid is, so back on the asconoid, this was a smooth, pretty smooth body wall, okay? In synconoid, that body wall gets folded. So it creates more surface area for choanocytes. So they generally can tend to get a little stronger of a current pulling them in. When you watch the video from Jonathan Bird, he'll show you how the current works through a sponge by injecting some dye into a sponge. Not injecting, spraying some dye at a sponge. The barrel sponge that he does, you see it really pull a bunch of the dye through, and that's because they have so much more surface area on which to have those choanocytes whip in their little flagella. So much water moves through them. Okay, so this is synconoid. They have this folded body wall, one atrium in the middle, and this guy right there, that is a synconoid body type. Okay, you can kind of see it's got the rougher body wall that's folded. Okay, again, if there is a professional sponge scientist, are you guys called porologists, by the way? If there's a porologist watching this, please feel free to correct me, please. Okay. These guys get bigger. So the picture on the first slide I showed you of the sponge that was bigger than the guy, that guy was Jonathan Bird. That is the synconoid body style. It allows them to get larger. They have more support. They can pull more water through. They can pull more food through. So they can build a larger body structure. Okay. Okay. There's again, giant barrel sponges. They're so cool. The third morphology is called leuconoid. These guys don't have one big atrium or one big spongiceal. They have a lot of little ones. So that's these guys, squishy sponges with lots of openings, lots of little chambers inside. Okay, so again, pause me. Go draw the leuconoid body type. It might take you another minute, so make sure that you label the osculum and the ostia in current canal. Okay, and again, this is leuconoid. So pause me, go do that. Did you do it? Okay, great. They have a whole series of canals inside of them, all lined with choanocytes, and these guys get really big. They take on a lot of different forms. They're much more free form. I've got these growing in my tank as an encrusting sponge. I've got these guys, these demos that I have for you. I've got little tiny guys. So these guys grow all over other things. So this sponge, which was gathered, I'm sure legally from a beach, this one actually has a clam shell, little bivalve shell, and a rock that it grew over and probably smothered. Poor little clam, oyster. Okay, so they just grow over and around and wherever they will fit. Okay, they can be huge and go on for quite a few meters. And again, they have, you can see the larger openings are their oscula, or one is an osculum covered with tiny ostia. Okay, this is just one example of a leuconoid style body. Okay. Whew, you guys, this is a lot of talking. You're doing great. Keep it up. Keep taking notes. Fabulous. Now we're going to talk about the taxonomy within the phylum Porifera. We're going to talk about 3 classes of sponges. Ready? Sponge taxonomy, class Calcarea. These are the only group of sponges that their spiculas are made of calcium carbonate. The ones that I know of, the ones that I've seen, are very, very small. These are pineapple sponges. They're very like prickly on the outside. They've got that calcium carbonate. They're kind of rough on the outside. They have a single osculum, so they are synconoid or asconoid shape. Maybe there are bigger calcareans. I've only ever seen tiny ones. I have these in my aquarium sometimes. They're hitchhikers. They just show up. And they've got this nice little furry collar around their osculum. Okay. Again, Calcarea, they have calcium carbonate, synconoid or asconoid shape. The second class is here we go. Hexactinellida. Remember me trying to say that word during the Prezi presentation? Hexactinellida. These guys are glass sponges. Oh, their spicules are made of silica, which is what we make glass from, and we make glass from sand. So a lot of the glass that we have might have once upon a time been a hexactinellidin sponge, and I've got one right here. Again, man, you guys, we got to come back to class so I can make you play with these things in real life. Okay, so you can see that. I mean, they look like glass. They're beautiful, and these guys all have asconoid morphology, so they're not huge, but look how intricate their body wall is, and just lovely. This is a Venus flower basket. That is a crab. This is a deep water, beautiful glass sponge. Okay. All right, last one, the class Demaspongiae. This is most of the sponges in the sponge world are demasponges, 5,000 known living species, and we're discovering more in the deep ocean as the people from awesome Mbari and Nautilus Live are down there cataloging the deep water life. These are leuconoid, asconoid, or synconoid sponges, some of the largest ones. These guys, their body walls and their support systems are the spongin fibers and silica spicules. Okay. And we have a local one of these. So this is Spongilla fragilis, and this is a freshwater sponge that is found in Utah Lake, so right there, you guys, I can almost see it from my window. Utah Lake and Salem Pond, and hopefully they're still there. Utah Lake has had some serious issues with toxic algae blooms for the last couple of years, which happens because we like to put fertilizer on our farms and our lawns. And all the streams that run down into Utah Lake carry that fertilizer with them, which makes the algae bloom. So there's been a lot of toxic problems in Utah Lake. So hopefully there are still some little Spongilla fragilis in there. It is a leuconoid sponge. They are tiny. I've got one right there. Okay. They're not dryable. They fall apart. But you guys we have sponges. Pretty cool. I think that's cool. Okay. Of course, I gotta have the weirdos for you guys. And first of all, let's look at this GIF up here. Yes, it's GIF. This sponge just sneezed. So I took a time-lapse video of a sponge expelling water quickly out of itself. So they're still sessile. They don't wander around, but they do respond to the environment a little more than we really thought. They're not just bumps. I mean, plants move, you know that, right? Like if you put a plant in a windowsill and you turn it, come back the next day, it will have grown, it's called phototropism. It'll turn and face the sunlight, right? So sponges move about that quickly too. It just takes a while. I mean, look at the timer on here, you guys. It's hundreds of minutes, 120 minutes it took this sponge to sneeze just to get crap out of itself. So they do move. And then also a couple of weird sponges. There are carnivorous deep sea sponges. This is an encrusting sponge right there. I will also link that video. And then this article I won't make you read, you're welcome, but it is about what's called a boring sponge. And it's not because they're no fun at parties. It's because they bore or drill into the animals' shells that they encrust. So this is an abalone shell. Abalone are a type of snail, and this shell, if you can see all of these little tiny holes all over it here, and they've actually gone into the internal surface of this abalone shell. So the sponge actually drills chemically through the calcium carbonate of the shell, and from what I've read, they don't actually do that to eat the snail's soft body. They do that, and it can cause the death of the snail or the clam that they do it on, because then bacteria can get in through those holes. Okay. Look at that. Look at it sneeze. It's crazy. Anyway, okay, I'm done. I'm so done talking today. Okay, your assignment for today, you need to upload pictures of your notes from this presentation, including your drawings of the morphology and water flow for a sponge, and then your Prezi for Porifera. Woohoo! You get to start building that map, right? You should have already turned in the first Prezi assignment, which was getting it set up, getting the characteristics of the kingdom Animalia done, and your title. If you haven't done that, why? Do it. If you need help, let me know. But for this week, you need to add a topic that will be Phylum Porifera. You're going to build a new topic section. You're going to list the basic characteristics of sponges. There was only 3 way back at the beginning of this very long video. And then you're going to add 3 subtopics to that Porifera. So when you're in your Porifera topic, you're going to go up to the top left, and hit subtopic 3 times, and each subtopic will be a class. You need to do Demospongiae, Calcarea, and Hexactinellida. And you are going to then list the characteristics and morphology for each of those classes, and then find an example species. And I've tried to make you guys practice this quite a bit over the last 6 weeks. Just the easiest way to do it is to type into the Google class Demospongiae example species. That'll pull something up for you. Okay, make sure you do common name and scientific name. Not all sponges have common names, so if it doesn't have a common name, don't stress about it, but make sure that scientific name, the genus and species, are formatted properly. They need to be, yes, italicized. Also, genus is right, capitalized. Species is correct, never capitalized. You might have to change the font. Some of Prezi's fonts don't italicize, so take your time, do your best, you'll be fine. Okay, that is worth 25 points, and it is due on, see, today is Monday the 12th, should be due on Wednesday, the end of next class. Okay? Okay. Next time I'll talk about Cnidaria. We're cramming them in. We're going group by group. Going. Okay? Okay. Oh no, wait, today's Tuesday. You guys are B day. Never mind, this is it for this week. Just this. This is your only assignment for this week. Okay? Okay, I hope you didn't stop it a minute ago. See, you should always listen. You should always watch to the very end, because you never know what Fravel's going to say, and you never know what Fravel's going to show you. Like, for instance, look who my TA is today. Daisy. I missed you guys so much. I brought my dog. Okay. Have a great week. Enjoy your fall break. Stay home because there's a pandemic. Okay. Talk to you soon. Wear your masks. Be good. Bye.